dead fish and so on and like that, and we isolated the fish here and found that they are antibiotic resistant. This has been published in many of these papers, and then of course we did with turtles. A very interesting thing is that we find turtles also containing the microbes originally from sewage and their antibiotic resistance, which is not supposed to be, because we are not giving to turtles or fish antibiotics. So that is very serious. It seems that in Oman, as I said, that I started the work while doing my graduate studies in 1985, 86, like that. And in fact, at that time, there was only 30% of antibiotic resistant from chicken. And now, whatever you're doing in chicken, you find 100% of the microbes are resistant to antibiotics. So we used it as bioindicators. The idea of biomonitoring and bioindicators, it was our own here to use this as bioindicators of pollution instead of the normal indicators. And of course, in addition to that, since you have sewage, then you'll have other things, but I wish that we can test all. You have pharmaceutical, you have other toxins and so on, but we did with heavy metals, and we can find that these heavy metals, we find them, again, it is in sewage effluent, but it is also in very similar heavy metals. You find it in turtles and turtle eggs. That's another indication that sewage is causing pollution. And these are the microanalyses that we did with the electron microscopy when you came and you visited the lab yesterday. So, what are the ways to do and to find out the toxicity there? We have the system there in our department where we can detect probably or assess that there is a kind of toxicity in there where we can record, and we use a specific, some aquatic animals to see that the behavior changes as analysis of that. That is very serious, because 2 years ago, almost there were, I think in 1 year time like that period, we have 4 outbreaks of the red tide. As you can see it here, these are the satellite images, and this is a mass kill of the fish here because of pollution that is going there. So where does it come, that pollution? It is a very serious situation, because in our country we know that we are in arid region, and these are the arid regions, as you can see. We are a part of it, and we know very well that we don't have enough rain, so we desalinate, which is very expensive. It goes to sewage, and that sewage, we treat it, then we use it for irrigation and so on. That has become very serious shortly, which is true. 2035 is not far away from now, but we're going to produce 70 million cubic meters of sewage. That is very serious. So here, we can see that in addition to sewage, there are also the compost that we are, this paper that came recently, and it is also containing the Omani compost, this, containing also heavy metals there. So we increase the pollution, heavy metal pollution in our country. So what is the solutions? There are already some institutions that have built like this situation where you can do phytoremediation using plants. These plants are not any, but there are some plants actually, they are even economically important, these plants, because they can absorb some heavy metals, and some actually can use, we have like precious metals like nickel and so on, to extract and probably use it for economical use. And the water that you get at the end is almost clean and so on, cleaner water, and it has been used. In these cases, this is just one of the pictures, but we use it even in terms of other big fields and so on for farmlands and so on like that. And usually phytoremediation, it is also involved the microbes in that case. That is very serious, because as we come, as we go, there is a declining of food production in Oman, and there is an increase of food import here. About 90% of our food that we import, we don't know. We don't even have actually a very good labs to analyze these foods. and to find out what is happening. So it is indirectly coming with all these things to the Ministry of Health for treatment and so on, because we don't know what is happening to the other side. So here, we find that our land have been damaged, and there is a lot of salts and so on like that. And we have additional water. So why we are not using that water also after cleaning it and so on to treat it? This is a paper published in Brazil, that they have very similar situation like that, and they use this microbe, Thiobacillus, which is common even here in Oman, to treat actually salinity. So there we are coming to biofertilizers, because the chemical fertilizers are very damaging. We use NPK, nitrogen, phosphate, and potassium, and it is actually going to underground water, and actually we end up having serious health problems there. So by using biofertilizers, probably we can solve these problems and so on. And we need the studies, and of course the people in Iran, they have some of these experience, as well as ourselves. There are some members in our department actually have published these papers, so the experience is there, but we need the exchange of knowledge and so on. So when we come, the other major problem is that, as we can see, one of the papers, I'll talk about it. But this is a common problem. I remember very well when I used to work in the Ministry of Health. We used to analyze for Staphylococcus aureus because of the enterotoxin produced by the Staphylococcus aureus and cause a lot of food poisoning and so on. So this is here, we did the NASA studies from the food handlers, and we collected these samples, and we isolate at least about these, identified about this number of enterotoxin in this type of food. The other major problem in our food is, in fact, especially now with the McDonald's, sorry, I'm attacking it, and some fast food, we eat a lot of fried food and so on, but we actually produce acrylamide, and we did this study. This was done in collaboration with Iran Pasteur Institute, but actually acrylamide is known very well to cause cancer. So here, the other very big problem is aflatoxin. We did a lot of aflatoxin study with the members, as you can see, different groups here, including even that we found it in turtles as well, but a lot of aflatoxin that are being imported in our food. And there is, I think this is here, we did it in Iran, okay? We presented that paper in Iran. But in addition to that, these are the few analyses, and I think we are ahead because we find probably the solution how to stop the microbes to produce aflatoxin and prevent health problem and intoxication. We used also gamma radiation to cure the food from aflatoxin. Our country, every time our doors are open, we don't have a very good quarantine, and we have introduced several diseases, one of the witch broom disease. As yesterday it was said, we used to have more than 1 million lemon trees here died because of the witch broom disease. But there is another one, probably you have seen it here in SQ, and we have published several papers. It is a new disease that is spreading very much in the plants, and now it is affecting, including some of the dead palms and so on. This is one of the few of the pictures that we have done it here. Now we got even somewhere, some places in Albatona that they have reported that. So we have to do biocontrol. We did some studies of the biocontrol, especially in Albatona area. They are suffering. There are a lot, about 35 parasitic nematodes killing our plants. And this is just some of the pictures, and these are the nematodes, the plant nematodes. But we did some studies of the biocontrol. This is our own study that we did it here, and we find that they are using chemical to treat this, and it is a failed one. They are resistant to chemical treatment, pesticides, and so on. So we use here biocontrol. These microbes, fungi, they are growing here. And actually, we tested these. We were able to grow it. And actually, they are 100% efficiency, at least at the lab scale, in preventing the disease. And I wish we can go ahead, but our number is so small to make like this work. The next step is actually biofuel, bioremediation, microbial enhancement, oil recovery. In terms of biofuel, we know that Oman now is looking, one of the strategies of the country is the biofuel. And in fact, the Department of Biology now, we have some members already being trained in terms of the biofuel. Dr. Lamia, that she was here yesterday, and Dr. Mohab also, they are actually for the biofuel. Dr. Lamia is the algal bloom. And of course, Dr. Nasreen from Iran, she's an expert in this particular area. They actually, biofuel studies and so on, and fermenters are actually in south of Iran because we have very similar environment as it is. We have high global competitiveness index, we have largest uninhibited area, long coastal line. All these are requirements for having a good biofuel from algal sources. So why biofuel? Because of the problems over the fossil fuels and so on like that. We have no choice to go to biofuel. We know how it is environmentally serious. And again, going back to health situation and going back to the Ministry of Health for treatment and so on. So here, this is from Dr. Lamia. She just graduated and published these papers about biofuel. And there is actually a vision if we can make biofuel production in Duqm or somewhere here. That is a point that we are looking ahead since it is one of the strategic plans. In addition to that, we are producing a lot of papers. And these papers actually, they are made of cellulose. And we got some microbes, very interesting also. One of these microbes can produce cellulase enzyme that will break down the cellulose. And we know that cellulose contain a lot of glucose that can be converted to biofuel. So here, that microbe, within 4 hours only, it can produce the maximum cellulase that was not reported again. So up to now internationally. So here it shows that these microbes break cellulase components and so on, and we look at the timing that they can do it, and here is the degradation where these things that we used, and they can actually degrade the paper and so on instead of being buried in landfills causing pollution or burning them. Actually, we can use them for production of biofuel. Biodegradation of the oil, we have isolated several microbes, and we also now we are very ahead in terms of will be related, as we're going to see later, to the oil industry. We know very well that about 35 to 40% or even more of the oil is passing through our coastlines here, and there is a lot of pollution from oil. And that's what you see here are the tankers. These are the super tankers, oil super tankers. And what you see, the black spots here, this is the satellite image, is in fact the oil that has been dumped when the oil tankers come to Oman here. Before they get into the Hormuz Strait, they dump the oil there from the balanced water and so on. And this is exactly what is happening. And of course, we have also a lot of desalination. We depend mainly on desalination, so imagine what will happen if there is a major break of oil and so on. We isolated the microbes. Very interesting that usually people say, okay, bioremediation is a slow process. But recently we isolate the microbes. It's only 14 hours almost, as you can see. This is the oil here that we put it in a petri dish, and within 14 hours that you can see that it is fully degrading almost that oil. Here is that you find is not much and so on. So we have isolated several of these microbes, but our champion is actually that one. When we come to, this will be immediately related to the MOR, microbial enhancement oil recovery. Because of the interest in Oman to do enhancement oil recovery, these are the components that usually used for the enhancement oil recovery. So what actually we can do is to look at these components and whether we can produce from the microbes or not. And the most expensive component is the surfactant that's used in oil recovery. Actually, if you look at in the metabolism, it's just simple. They want these to make the cells and biomass, biopolymers, biosurfactant, all these components in breaking down of the nutrients and so on. We have published several papers in this area, and we are very successful, very competitive internationally. And in fact, even isolate, we have now 4 technologies in our hands. They are actually to be released and to go to the field to be tested. But this is the surfactant that we have isolated. It is unique of its kind because it resists high temperature, pH, and salinity. So it is a very good one. And these are just some of the microbes that we find here in Oman. And we can see that when we mixed that biosurfactant with some chemical surfactant, we are able to recover about 50% of additional oil at the lab scale. So it is, and we did, by the way, I did not add it here, but we did studies, economical studies related to this, and it is very economic because even if we dilute it 20 folds, still our biosurfactant is active. We did, one of the major issue here is the heavy oil. So also we try to get the microbes for the heavy oil to break it, to make it lighter oil. So we got some studies, the heavy oil that with the C70, C80 chain, we were able to use some of the microbes and break it between C11 to C27. Some of these microbes are actually among those. So here it was actually our approach is to contact, we were asked after the several successes of the microbial enhancement recovery, we were asked to make a lab, and the petroleum company will provide us with the machineries, and we have to make that lab. So the lab is actually specifically not only for the oil, but as you can see it, we have even here the bioreactors. And the bioreactors where we need people from Iran to help us to put this. When we start to make the bioreactor, then we actually open a new era in Oman for production, because if you have a bioreactor, the same that bioreactor, you can use it for training people to produce biopharmaceuticals, a lot. We talked about biofuels. From the algae, you can produce anti-aging, anti-cancers, and so on like that. In addition, we can produce even food materials like vinegar and so on. Our country, before our grandmothers and so on, we used to make vinegar at home and yeast at home. But now, sorry that we are actually just consumers. We are not producing anything. We buy vinegar from somewhere else instead of producing it our own, and the technology is easy to make it. So we need actually to have like this lab. This is the actual plan of that lab. So the outcomes summary of this, we actually have indigenous technologies in our hand, indigenous microbes from there. We have the biomass, biosurfactant, biogas, and I forgot to add something else here that we were asked to also do biopolymer for this, and we are successfully doing it. We are in the middle of the way. So summary and conclusion, I feel these are the collaboration points where we can collaborate, and probably whether issues related to antibiotic resistance are bioindicators or from the hospitals and how they get there, and how actually we can find out additional genes, because if you look at tetracycline itself, only the tetracycline you have 40 genes, and we have identified only 7 so far. So for sure we need this, a big scale to do that study. We need to use pollution indicators. We use antibiotic resistance, heavy metal, probably pharmaceutical components and so on. Microbial toxin in food, secondary metabolites, as I have showed before, biofertilizers, biofuel, biodegradation of environmental pollution, and of course, microbial enhancement oil recovery. And as yesterday we visited the Central Analytical and Applied Facility, in the College of Science, we were talking also the expense to have these diagnostic kits. Can we collaborate with the Iranians to make our own diagnostic kit in a cheaper price instead of getting it abroad? Well, that's the whole thing, and thank you very much.
Thank you, Professor, for the very interesting and informative presentation. Now the door is open for questions. Yeah, Prof. Nasreen.
Doesn't work. I think the other one, it works. Doesn't?
I asked a question about the resistance to antibiotics. In Iran, people easily they can find antibiotics from the pharmaceutical without any prescription of the medical doctor. And they are using a lot of the antibiotics as growth factor in dairy production, dairy industry, for the animal feed, and also in the chicken. There is same condition in Oman?
Yes, it is. It is. The Ministry of Health actually has put strong regulations about this, but the issue, a lot of our regulations, do we follow them? That's the question. I think even Iran, you have probably very good regulations, but we don't follow it. There was a very interesting case while we were doing this study. We went to one of the chicken factories, and we found that actually they throw tetracycline just like that. They give to animals just like that. That is very serious situation. And in fact, I remember while I was working with some national committees, we were informed while I was talking about this and find the solutions of this, that there was one factory gave chicken a lot of antibiotic until they went crazy. They were chasing whoever is getting inside, and the owner of that was told, execute all these chickens because they are not healthy and so on. So the issue of our antibiotics is a very serious situation. And I think, yes, I don't think the Ministry of Health by themselves can do it. I think we need a very strong collaboration. We need, although there are very good regulations, but now we need actually to execute these regulations and to find out exactly what the cause, because with the WHO recommendation, as it is very worrisome, it seems that you are facing a very serious situation, and they even say it will look like the pre-antibiotic situation if we don't know how to solve it. In fact, very recently, a lot of antibiotic resistant, even E. coli that our normal flora now can cause septicemia, can cause meningitis, the Mycobacterium tuberculosis now in many places, including Iran, to my knowledge, is spreading very fast. In fact, very sad, in Oman, although SQ, we have done these studies and others studies that have been done, but if you look at that report, there is only one line of 1,300 resistant microbe, which is also 65% resistance. When you come to Iran, you find even urology and other places and so on, they reach about 70%. In all cases, I think we really have to work together about this issue.
Yes. More questions, please.
That's, I think, the very, very important thing. I was just talking to Dr. Yahya, not only for this, but for the whole team, whether we are talking about the health issue with hospitals, or whether we are talking about the petroleum, like we did yesterday, or nanotechnology, or biotechnology, that actually we need a very clear plan for the collaboration. One of the issues that actually all we talked about this, but we did not put it, including myself there, is exchange of faculty members, exchange of students, exchange of samples, and so on like that. What is the strategy? What is the next way forward? That I think we need to all to sit together and find a way to make this collaboration successful and to find exactly how we can do that.
Okay.
Excuse me. Is this bacterial resistant only to tetracycline? Because tetracycline is not clinically popular to use in clinical medicine today. So what is the
Okay, thank you. That's an interesting question because I faced a very similar question from some medical doctors. Tetracycline, we did with 15 antibiotics, but I'm reporting here tetracycline because it's one of the recent studies that I went and did even genetic studies with my students. That's the reason I'm reporting it. But if you look at streptomycin, if you look at gentamicin and other antibiotics, they are resistant. The issue of many of these antibiotics are broad-spectrum antibiotics. Usually when you use a broad-spectrum antibiotic, for us, a medical doctor, you are trying to help a patient before the crisis is coming. But actually we promote antibiotic resistance because of the broad spectrum. They make resistant to other antibiotics as well. So it is so important that before prescribing some of these antibiotics to find out, probably there is no time from a doctor point, but it is better every time that to look at to do antibiotic resistance studies before we prescribe and follow up that system. Many people internationally, especially when a long-term antibiotics like for Mycobacterium tuberculosis treatment is a very serious issue because it is a long-term and expensive. So people cut in the middle. The 5 days prescription the doctors give, there are many people, they cut it before even completing the 5 days. But that is what caused all these problems as a part of it. And when, of course, the sewage is a meeting point because you have whatever, chicken and so on and like that, and then you have from human sewage and so on. The sewage treatment plant is a meeting point of exchanging the genes. The one that I reported here with 4, it was in sewage. But similar situation we find in chicken and human beings.
Excuse me, there's no time, but the problem is if you look in the guidelines, clinical guidelines, unfortunately, many of infectious diseases in urinary infection and so on, still they put antibiotics and in all surgeries and medical legally. If some doctors don't use antibiotics, if something happened, they say, oh, why didn't they use these antibiotics? Even evidence shows that only before surgery you give antibiotic, that's quite enough. You shouldn't continue after that. But everybody follows that because of these legal problems, and this is an international problem, as you mentioned, and there's no solution in short term now.
Yeah, it's true you cannot avoid antibiotics, but at the same time, if you look at in Europe, for example, they ban the use of antibiotic as growth promoters in animals. If you come to many of our countries, one of the papers, the one that I'm reporting there, was sent to a reviewer in Europe and said, we have banned it. What about your country? And I said, no, we haven't. And I brought evidence, not only in the country, in our country, but if you go to Iran, India, Pakistan, South America, even in the US. So the problem of mainly it comes from the earth growth promotion, especially when it is a broad spectrum, it creates all these problems. As a medical doctor, as I said, in many cases you have no choice. You have to use it. But what will happen next? That is why we produce first generation, second generation, and I think now we are at the 4th generation. And they isolated, I think about 6-7 years ago, Staphylococcus aureus with 99 antibiotic resistancy with the exception of vancomycin.
Yeah, Prof. The time is over, sorry.
Yes.
Time is over, so, yeah, please.
I don't want to take your time too much, but I guess maybe Dr. Yahya also help us. Actually, at the beginning, I should appreciate the talk. I was access to your, vast type of the research that you have done, and that's a good model, I guess, for the whole department in the SQU that you have already collaboration with each other. Okay, so and I'm very optimistic also to have collaboration with Iranian universities and institutes. Regarding the MUR, Doctor, have you ever done any screening on in Omani reservoirs to see if any of those are suitable for MUR or not because of the salinity and temperature?
Yeah, we did, but not for all Omani reservoirs, definitely, just for one reservoir only. Yeah, so we did that. Yeah.
Okay.
Time is over, actually. Sorry, Prof, because.
We have the genetically island that they can migrate it between the microorganisms in environment, in water, in human, and all the living organisms. And this is an interaction between the gene and environment. So even if we are not using an antibiotic for the treatment in the clinical use, but this island of the genetically mutation, they can move from environment to the microorganism that they are pathogen. And the resistance, this is the reason that is distributed totally worldwide.
All right,
It's,
okay, thank
it's
you.
exactly what
Thank
I have showed at the first slide. You can't avoid and say just clinical and avoid the use of growth promoters or avoid the environment. It's coming back to us.
Okay, thank you, Prof.
Thank
I
you.
already made wasta for you and gave you more than. Okay, so our next speaker is Dr. Masoud Hoshman from Masoud Dan. All right, so yeah, all right, okay. Then Professor Nasreen Muazzami from Iran, from the Iranian Research Organization for Sciences and Technology, IROST. Her presentation will be about biofuel production using microalgae. Please, floor is yours. All right, thank you. Thank you so much. Thank you. That was excellent, but you took half an hour more, maybe, the whole session. Yeah.
Permission of the President and my colleague. I'm talking about the microalgae as the first biorefinery establishment in the south part of Iran. And I mention biorefinery because we can produce a huge different product from the fuel to medicine, pharmaceutical, biofertilizer, bioplastic, and many molecule that we are not identified now. The project focus on two main areas. One is the replacement of the part of the fossil fuel, and second one, sequestration of the CO2, because Iran is number 7 in the 10 first country that they are produced and they are emitted the CO2. And in the near future, we should be paid many penalty for this problem. And the third one, the most important fuel and biofuel for the future is the fuel for transportation. So we need the liquid fuel for the replacement of fossil fuel for transportation. And how does algae grow, and what is the need of the growth of the algae is the sunlight, is the water, is the land, and carbon dioxide. Normally, the microalgae, they are sequestrated the CO2 from atmosphere, but the concentration of CO2 is not very high, it's around 0.3. And when we are added or we are diffused the carbon dioxide in the cultivation system, we can increase the efficiency of the photosynthesis and produce large quantity of the biomass. 1 kilogram or 1 gram of biomass of the microalgae, they can sequestrate 1.8 CO2. The background of the project began 14 years ago. We are isolated 107 47 microorganisms, microalgae, and we are selected in first screening step, the high producer of the biomass, and in this next time or next step, the microalgae that they are produce high quantity of oil, and all the microalgae they are produce the oil, but all the oil cannot convert it to the biofuel. There is some standard determination of the carbon chain, and this is the kind of the oil of microalgae that they can convert it to the biofuel. Just some picture of our background of the project. The microalgae pilot plant in 2002, sorry, this is different place. This should be changed, the place of the pilot plant. I'm establishing Qeshmai land, a closed system and open system, and this is the pilot plant existed in Irost in Tehran with the capacity of 25,000 liter production of biomass. And for the production of the oil, as I mentioned, we need only the light, the sunshine, CO2, water, and the land for large-scale cultivation. And why we are using the microalgae? This is comparison between the oil seed production of oil and the microalgae is not compared. And secondly, for the cultivation of oil seed, we need the agricultural land and the fresh water. Microalgae can grow in seawater, and they don't need the agricultural land. This is a picture of our pilot plant in Tehran. Another picture of pilot plant. You can see the open pond for the large-scale cultivation of microalgae. This is the concentration of the oil in the cells, the selected organism that we are developed for large-scale cultivation. By light microscope, you can see the globule of the oil concentrated in the cell. From the 147 microalgae we are selected for because of the specificity of the oil and also the high productivity of the biomass and oil. And we are deposited in Persian type culture collection. The first one produced 72% of oil per biomass, dry mass. And this is the profile of the oil. As I mentioned, some kind of the profile they can convert it to the biofuel. Regarding this standard, a worldwide standard, is the carbon chain between 14 to 24. Less than 14 is not suitable for the conversion, and more than 24 carbon is the same. When we are talking the biorefinery, in a refinery of the crude oil, we can produce the diesel, produce the jet fuel, gasoline, different kind of petrochemical product like alcohol, fertilizer, chemical fertilizer, and plastic. But with the biorefinery of microalgae, we can produce all of this product. We can produce the polysaccharide, we can produce the enzyme, pigment, mineral, the vitamin, essential oil, antibiotics, antiviral, and vaccine. We can cloning the gene in microalgae for production of vaccine. I mentioned about our study that is established now in Iros. And the most important, this technology is environment manager. We can control CO2 emission, we can sequestrate CO2 and reduce the problem of global change. This is the carbon chain that they can convert it to different kind of biofuel. For the gasoline, this is the carbon number, and also boiling point is between 4 to 12. For jet fuel, between 8 to 16, and high quality diesel is between 8 to 22 or 20. In the biorefinery, we can produce the food and feed, fine and bulk chemical, fiber and biomaterial. Two companies existed now in Italy and the USA that they are produce the bioplastic from microalgae, fuel and energy, And biofertilizer, many of microalgae, they can sequestrate of nitrogen and phosphorus in the cell. And 1 ton of this kind of biofertilizer, that's enough for 30 hectares of the agricultural land. Our product and our technology that we have developed between 2002 to now is the biofuel, the biojet, gasoline, and high-quality diesel that I introduced the technology. And the second part is the anti-aging product. This is a collaboration, close collaboration with Medical Council of Iran. Medical Council of Iran, they are established regarding the Millennium Goals 2000, the national sustainable health for the country. And the goals and the aims of this center is to prevent the chronic disease of the people over 65 years and the future generation of Iran. In this category, we are produced a nutraceutical enriched by iron, calcium, selenium, is necessary other mineral for the micro deficiency in the people. That is very high in Iran. The therapeutic protein is the protein from microalgae. We are using the enzyme to separate it, the different kind of amino acid. The phycocyanin, powder of dunaliella, as a source of provitamin, and this is a contract with the World Food Program to add the vitamin